Mass General Brigham Innovation Discovery Grant Program, IDG, began in 2014 and is now in its 7th round of funding. To date, $4.2 million has been awarded to 65 projects. 14 new companies are actively developing IDG-supported technologies, with 19 license agreements executed to further their development. And more than $199 million in follow-on funding has been received to further development of the IDG-supported technologies, a 47-fold increase on the funds awarded. The IDG program provides funding and access to resources for the purpose of translating academic discoveries into commercial products to make them available to physicians and patients to improve human health. The program supports researchers who have developed technologies using traditional sources of funding and enables them to pivot towards commercialization of those technologies by supporting access to commercial development expertise and resources. Please join us in congratulating this year's IDG awardees. Miles Cunningham, MD, PhD, at McLean Hospital, a novel convection enhanced delivery system for brain malignancies. A direct delivery system for infusing a therapeutic agent within diseased brain tissue to treat glioblastoma multiforme, or GBM. The project uses sophisticated surgical planning software and rapid prototyping to fabricate a patient-specific system within 1 to 2 days of diagnosis. Hilary Gallin, MD, MBA, at Massachusetts General Hospital, FastLine single-handed venous access device. A device that enables placement of a central line with one hand using intuitive, ergonomic movements to enable real-time troubleshooting and allow the use of ultrasound in the other hand throughout the entire procedure. The device could reduce complications and related costs, decrease procedure time, and improve the patient experience. Vidyasagar Koduri, MD, PhD, at Brigham and Women's Hospital, development of KRAS degraders in cancer. Mutations in the KRAS, Kirsten rat sarcoma virus gene, are the main drivers of lung, colon, and pancreatic cancer, which combined kill 230,000 annually in the U.S. The proposed research program uses a newly developed novel positive selection screening platform as a tool to identify protein degraders of KRAS as potential therapeutic agents. Arpita Kulkarni, PhD, at Brigham and Women's Hospital, restoring tumor immunogenicity in glioblastoma multiforme, GBM, a novel drug that could change the paradigm for glioblastoma or GBM therapy, which has remained unchanged for decades. The program is engineering a new therapeutic that can cross the blood-brain barrier, induce tumor immunogenicity in models of GBM and other solid cancers, and synergize with cell therapies. Lee Lan, MD, PhD, at Massachusetts General Hospital, developing first-in-class mRNA methyltransferase inhibitor, targeting a newly identified mRNA-dependent repair pathway and mRNA methylating enzymes in DNA repair in cancer therapy. This could lead to effective therapeutic strategies for around 90% of breast and ovarian cancer patients without HRD, as well as effective therapeutic strategies for other cancers. Klaas Lindman, PhD, at Spalding Rehabilitation Network, auditory mirror therapy for tinnitus. This proposed technology provides a new type of treatment for tinnitus that is non-invasive, affordable, and low risk. Based on disruption of multisensory integration and implemented in a pair of headphones, a pilot trial of this auditory mirror therapy indicates significantly and substantially reduced tinnitus. Hamid Matalebzadeh, PhD, at Massachusetts Eye and Ear, non-invasive diagnosis of middle ear pathologies. Approximately 15% of American adults, or 37.5 million, aged 18 and over, report some trouble hearing, and over 50% of these cases are related to conductive hearing loss. This project aims to automate objective differential diagnoses of middle ear pathologies by using powerful machine learning pattern recognition to infer details about middle ear status from clinical data. Jin-Mao Park, PhD, at Massachusetts General Hospital. Small molecule MMP inhibitors for allergic inflammation. Current allergy medications are effective in only subsets of patients, produce partial responses, and often encounter treatment resistance. More importantly, they can directly interfere with the immune system and cause harmful side effects. This project aims to develop small molecule inhibitors of a specific protein-degrading enzyme as topical therapeutics for atopic dermatitis, asthma, and other allergic inflammatory diseases. David Pepin, PhD, Massachusetts General Hospital. Neutralizing antibody to FSTL3 as an immunotherapy in ovarian cancer. High-grade serous ovarian cancer is the most lethal gynecological malignancy, accounting for 70% of ovarian cancer-related deaths. The new technology consists of a neutralizing antibody that would be used in combination with immune checkpoint blockade therapy and alongside the current standard of care for ovarian cancer. Hope U, MD, Brigham and Women's Hospital. Circulating microparticle proteins predict pregnancies complicated by placenta accreta spectrum. Placenta accreta spectrum, or PAS, is a potentially life-threatening pregnancy complication that occurs in approximately 1 in every 1,000 to 2,000 pregnancies. The proposed product is a blood test to assess women's risk using circulatory microparticle proteins, or CMP. This test would be used at the end of the second trimester in routine outpatient blood work to improve the antenatal detection of PAS. Congratulations again to this year's IDG awardees.